Okay, very good evening, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So today's our eminent resource person, Dr. D.R.G.BHAGIRATH Garu, Assistant Professor, Head Department of Botany, Government Degree College for Rangasaipet. So, sir is very expert in molecular biology, especially plant genetics. So during the class, if you have any doubt, please ask our respected resource person. And also, please practice whenever we are sending the assignments or the math tests in our WhatsApp group. We have already prepared each and every topic. We are already prepared. So each and every topic, we are already prepared assignments and the quizzes also. So, my dear friends, I have prepared and previous papers, what we have already, the people, those who are written in previous CPGET 2022, 23 or 21, like that, the previous papers we have already analyzed. And that video also uploaded in YouTube. That video links also sent in our WhatsApp group. So during in your free time, if you have any free time, please make sure listen that videographs and video. And if you have any doubt during listening the video, please make your own questions and also the make notes, your own notes. That is very helpful to you. And regarding the CPGET, or if you have any doubt of that video or the content, you directly interact with me. You ask me. Definitely, I will interact with you, my dear friends. No doubt at all. And also, the people, those who are not yet applied for CPGET 2024, don't be delay, my dear friends. I am suggesting you, please you apply your CPGET 2024 online application. So already Usmania University website CPGET 2024 online is open. I have sent the links in our WhatsApp group also. So it is very easier to search in a website. So I have sent the links. You can click the send, click the links, and directly you have to enter into the website and apply your CPGET 2024 botany entrance examination. So whenever you are first come first serve, so that's why you should apply earlier and make sure you going to opt very nearer to the your native place, that is examination center. It is an online question paper, so that's why it is very helpful to you. Don't be delay, my dear friends. So don't be delay and rush immediately. By today or tomorrow itself, you can go and open the website and directly apply, online apply. And after completion of the application, you should send your online application also through my WhatsApp. Okay. So I would like to invite today's our eminent resource person, Dr. D.R.G.BHAGIRATH Garu. Please welcome and share your screen and start your session, sir. Please welcome.
Thank you. Thank you for giving this opportunity. And also, I wish you all life science students. Today is a very auspicious day. That is the International Biological Diversity.
Yeah.
So we are all are the part in the save the biodiversity of our country as well as worldwide phenomenon. Okay. Shall I now share my screen?
Okay. Please proceed.
So I think it's visible.
Yeah, please enlarge.
Okay.
It is visible. Please enlarge.
Okay.
Okay.
Okay. Today we are take continuation part of mutations. So mutations are nothing but it is a sudden change, whether it is a qualitative change or a quantitative change occurs in the genetic material of the organism and which transmit to the next generation. So the mutations are broadly classified into major three categories. One is chromosomal mutations. We already completed that part. And today we'll discuss about second part of the mutations, that is gene mutation or point mutation. Sometimes it is called factorial mutations or micro mutations. So these are the different names are given to that. Why? Because our genetic material is made up of with millions of nucleotides, everyone knows. So gene is nothing but it is the smallest part of the DNA or a tiny part of the DNA, which manifest a particular character in the organism. So a small change leads to it's a change in the gene product or change in the type of metabolism, or a particular biochemical reaction is also altered, or sometimes the character is also changed due to a small minute change. That change occurs within a small nucleotide. That is nothing but adenine, guanine, cytosine, thymine. Among the four nucleotides, one of the nucleotide may be replaced with other, or may be deleted sometimes, or sometimes add a new nucleotide into the genomic material. It leads to getting a new character in the further generations. So these gene mutations are again we can classify it into different way. So where it is the mutations are occurs, based on that it will be divided into two types: somatic mutations and germinal mutations. What is meant by somatic mutations? Those mutations are occur in the somatic cells, just like root cell or the stem or the leaf cells, like that. So శారీరక భాగాల్లో జరిగే ఉత్పరివర్తనాన్ని మనం సొమాటిక్ మ్యుటేషన్స్ అని చెప్పి మనం పిలవచ్చు. సో వేర్ ఇట్ ఈస్ అక్కర్స్, ఉనికిని బట్టి ఆర్ ఆరిజిన్ ఆఫ్ ద మ్యుటేషన్స్, వేర్ ఇట్ ఈస్ అక్కర్స్, it's divided into two types. One is somatic, another one is germinal. Generally, the somatic mutations are restricted to a particular generation only. It may not pass to the next generation. If it is employ different vegetative propagation methods, definitely it also continue in the next generations, like vegetative propagation methods like budding or grafting, or some other methods are there in the plants. So example is golden delicious apple, navel orange, and emperor seedless grapes. These are all best examples for the somatic mutations. So when these mutations are occur in the flower bud is also, we can call as not flower bud, it is vegetative bud. We can call as bud mutations. So example is buntark, fontiac, cobbler. Different type of mutations are observed in the potato. So these mutations are continue to the next generation if you employ the different type of vegetative propagation methods like that. So these are the called somatic mutations. And next, another type of mutation is called as germinal mutations. Germinal mutations is nothing but at the time of gametogenesis, or sometimes in the zygote cell or embryo also. These cells are responsible for the change in the characters of the organism, which transmit to the next generation. So we are called that the wild type of genes are converted into mutant type of genes. So these mutations are called as germinal mutations. So again, it is based on that whether it occurs in the zygote or the gametes. You can call gametic mutations and zygotic mutations. Example, Headford cattles and gray-headed what is that cattles. These are the good examples for the germinal mutations. And another one is spontaneous mutations. Spontaneous mutations means which occurs without influence of the human beings. Naturally, mutations are seen in different organisms due to environmental factors or internal factors of the organism. Maybe the genetic material undergo some changes. It leads to mutations, but the spontaneous mutations are very, very less frequency in crop plants. So it is moreover, it is difficult to identify the spontaneous mutations in the crop population. ఒక పంట జనాభాలో ఆకస్మికంగా సంభవించేటువంటి ఈ మార్పులను గుర్తించడం కూడా చాలా కష్టం. వీటినే మనం యాదృచ్ఛిక ఉత్పరివర్తనాలు లేదా స్పాంటేనియస్ మ్యుటేషన్స్ అంటారు. ఎగ్జాంపుల్ ప్యాడీలో GEB-24 వెరైటీని డెవలప్ చేయడం జరిగింది ఈ మ్యుటేషన్స్ ఆధారంగా. అదేవిధంగా EC3735 రాగులలో వీటిని ఐడెంటిఫై చేయడం జరిగింది. దట్ ఈస్ కాల్డ్ స్పాంటేనియస్ మ్యుటేషన్స్. Based on the direction of the mutation, we can call as forward mutations, and another one is reverse mutations. Forward mutations are పురోగామి ఉత్పరివర్తనాలు అంటారు. పురోగామి ఉత్పరివర్తనాలను మనం ఎప్పుడు పిలవచ్చు వీటిని అంటే ఏదైనా ఒక వైల్డ్ జీన్ అంటే వన్యజన్యు అనేది మ్యూటెంట్ జీన్ గా మారినప్పుడు అంటే వైల్డ్ క్యారెక్టర్ ఈస్ కన్వర్టెడ్ ఇంటూ ఏ డిఫరెంట్ ఫినోటైపిక్ క్యారెక్టర్ ఆర్ మ్యూటెంట్ టైప్ by changing in the genetic material. Sometimes the mutant gene is also reversible into wild one. So that is called reverse mutations. అంటే కొన్ని జనరేషన్స్ వరకు ఆ క్యారెక్టర్ మెయింటైన్ అవుతూ ఉంటుంది. సడన్ గా ఒక తరంలో మళ్ళీ ఆ క్యారెక్టర్ అనేది రివర్స్ అంటే పూర్వ స్థాయికి ప్రాథమిక స్థాయి క్యారెక్టర్ లోకి వెళ్ళిపోతుంది అనమాట. ఫార్వర్డ్ అండ్ రివర్స్ mutations. The wild gene may be converted into mutant gene or mutant gene is converted into wild gene. So this is based on the direction of the mutation. We can call forward mutation. Another one is reverse mutations. So in the during the development of the scientific technology and hybridization methods, the human beings also try to evolve new varieties or good varieties among the crop plants. Try to induce the mutations in the crop plants by using different type of mutagens. Mutagens are nothing but chemical compound which influence the or which alter the genomic sequence of the original genomic sequence of the organisms. So these induced mutations inculcated by applying different chemical agents, whether it is physical mutagens or chemical mutagens. Physical mutagens means that is ionization radiations like alpha, beta rays, UV rays, X-rays. These are the different type of radiations are used for the causing mutations in the crop plants as well as the animals also. Example is Stadler is there. గోధుమలో ఇతడు ఉత్పరివర్తనాలను సాధించడం జరిగింది X కిరణాలను ఉపయోగించి. By using X-rays, Stadler introduced the new varieties by applying X-rays on the wheat plant. So that is induced mutations. So chemical mutations also రసాయనిక ఉత్పరివర్తనాలని మనం చెప్తాం. So by using different chemical agents like HNO2, mustard gas, formaldehyde, colchicine or other chemical compounds also, by using these chemical compounds which change the genomic material, which changes in the genomic material, we can call as chemical mutagens. So chemical mutagens are different types are there. So example is the nitrous acid. So and most of the times nitrous acid is used in Aspergillus fungi, and mustard gas which is used in the male Drosophila insects, and which shows more highest rate of mutation, but experimentally it is proved. These are all different type of chemicals which used for the formaldehyde, caffeine, manganese salts, which are used to in the bacteria, which causes mutations in bacteria also. These are called induced mutations. So physical mutagens and chemical mutagens. So in the physical mutagens, ionizing radiations are there, non-ionizing radiations are there. So ionizing radiations are X-rays, that is called alpha rays, beta rays are there. Non-ionizing radiation is UV, visible rays are non-ionizing radiations are there. So this is about the different type of mutations. Next, I will share another PDF file. Just go through that one.